Our final discussion about speciation is going to talk about the barriers that create separation between interbreeding populations. This is critical for speciation. Now remember, inevitably, if we have a barrier, then there will be speciation, but it usually takes thousands and thousands of generations. So if at any point the separation between those two populations breaks down, the process of speciation could reverse and we could get that population mixing and all of the genetic diversity that was in population 1 being spread to population 2 and vice versa, so it would be just like one big population. Now, we're going to divide these barriers into several different categories. In fact, there are 3 that you need to know. For species that have been separated a very long time, all of these, in fact, many individual subcategories under each of these major categories will be in place. My funny, kind of silly example is, imagine you wanted to try and breed an iguanophant, half iguana, half elephant, right? You can make $1 million selling them as pets. You realize, though, hopefully, of course, that that's ridiculous. They have been separated so long that there are many, many, many barriers. So let's look, and we'll look at some examples here under each of these categories. The first type of isolation is a premating barrier. Now, geography obviously can be a premating barrier. If you can't get to one another to mate, then that's going to be a premating barrier. And so premating barrier is anything that prevents or greatly reduces two individuals' opportunities to get together and mate. So habitat could be this. If they were living in the same area, but one was down in the forest floor and one was up in the canopy and they very rarely moved back and forth, those different habitats would be a good premating barrier. Behaviorally, we looked at the fruit flies and how they were niche partitioning on those different trees, the crab apple trees and then the domesticated apple trees. That's kind of a behavioral, maybe you can even think of it as a little bit of a subhabitat barrier. And then temporal ones are ones that are for timing. So diurnal versus nocturnal is a type of a temporal premating barrier. One of my favorite premating barriers is cicadas. Cicadas are insects that in the summer here in the south, they come out and they make that large kind of loud, high-pitched squealing, screaming noise up in the trees. Sometimes it's really, really loud. Well, they're actually, depending on where you live, 2 or 3, sometimes even 4 different species, but they're on different cycles. Some of them take as long as 13 or 17 years to go through the process of living underground. They drink the sap from roots of trees, getting enough energy to where they can finally become adults and mate. And so if you've got a 5-year cycle species and a 13-year cycle species, even though they're in the same area, they very rarely are emerging on the same year. So we'd have to look at whatever the common product of 5 and 13 is. So maybe once every 100, 200 years they emerge on the same year and would have the opportunity to mate. But other than that, they wouldn't. So those are all pre-mating barriers. Now in flowers, as we have pictured here, many flowers have pre-mating barriers based on their pollinator. If they have a very specific pollinator, only one insect that is, or primarily one insect that's pollinating them, like if the hawk moth only pollinates flower A, then that's going to be a pre-mating barrier. Won't have the opportunity, or very rarely, for pollen to get from this flower to this one or this one. So different pollinators can be a good pre-mating barrier in plants. And sometimes if we're going to define species using that biological species definition, remember that's one where we base species boundaries on whether or not they can exchange genes, then in some cases a extreme pre-mating barrier might create what we may define as an instantaneous species boundary. And this has happened multiple times in snails. So snail species typically have their shells that only go in one direction, but different species might have a different direction. So this is sinistral, meaning left, versus dextral, meaning right. Now notice both of these are dextral. They match this blue one here. If you flip this one over, it would match. And these species of snails are most commonly hermaphrodites, but they do need a mate. They can't self-fertilize. So these snails, it's a very interesting mating system. They'll line up male and female reproductive tracts. There'll be a two-way exchange of sperm, so each one will donate sperm to the other one, and both of them will crawl away pregnant, right? Their female reproductive parts will be fertilized. However, if a dextral snail tries to mate with a sinistral snail, they can't line up. The shells just get in the way. Without getting too graphic, their shells make mating nearly impossible. So if we have, as in this red lineage here, an entirely sinistral lineage, but then there's a rare mutant that's dextral, most of the time that mutant's out of luck. He can't mate with any other individuals in his population. He will die without having, he, she, right, hermaphrodite, will die without having any offspring. However, very rarely we have two mutants that occur and they find each other, and they're able to mate just fine because they both have right-handed shells. So if that rare thing happens and they establish a right-handed population, right-shelled I guess I should say, then that can be a brand new species. And so in theory, that one mutation event, like really, really rapidly, could represent a speciation event, even though they're still almost completely genetically identical except for that one change. It would, by a biological definition, right, are they able to mate, would be a new species. So this type of rapid reproductive isolation can, in extreme cases, be very quick speciation using a narrow definition. Okay? But all of those are pre-mating barriers. They all prevent or at least greatly limit the ability of individuals to mate. Now, if mating can occur or is attempted quite readily, but there's something downstream of mating, so between mating and the zygote, we call it a prezygotic barrier. So mating can and occurs quite frequently, but a zygote never forms. So one example of this is in flies. So these are actually fly male reproductive structures. Sorry, this is fly pornography, but hopefully it's not too offensive to you. And each of these will fit the female reproductive tract for their own species, but they do not fit the female reproductive tract of another species. And so they will mate, they'll go through the pre-mating, sometimes there's a dance or just sizing one another up, and they'll attempt to mate, they'll go through the mechanical motions of mating, but it's never fully realized because sperm just cannot, they're very, very difficult to get it transferred. So that creates an effective barrier, but it's not a mating barrier, it's a barrier from the zygote forming. And in broadcast spawners, so broadcast spawners are organisms that release their eggs and sperm up into the water, and then fertilization occurs out in the water column. Corals do this. And so different corals have maintained their species diversity by having eggs and sperm that cannot fuse with one another. Now realize, back to our iguanodont example, let's say I was really serious about making a hybrid between an elephant and an iguana. So I do a artificial insemination or an in vitro assimilation, in vitro fertilization. I get eggs from an elephant and sperm from an iguana. I put them in a test tube and mix them together. They're also going to have this gametic incompatibility, right? Iguana sperm won't recognize elephant eggs. That's actually one of the most rapid things that changes after species diversify. So gametic incompatibility is really important for broadcast spawners, but it happens in internal fertilizers all the time too, right? So a tiger sperm will actually still recognize a lion egg. So they have not developed that. There's some other barriers which we're going to talk about next. But before we do that, I'm going to give one more extreme example of a post-zygotic barrier. And in this one, mating occurs. These have a very generalist pollinator, so pollen is easily transferred back and forth. However, in many plants, they go through polyploid speciation, which means they have a mutation where they have extra chromosomes, so maybe an entire extra copy of their genome. And when that occurs, if it occurs in an odd number, so if you have 3 copies, usually it means you're completely unable to make pollen or eggs. And so those individuals can't reproduce via fertilization. They may be able to reproduce by sending out runners or from the roots or other asexual ways that plants reproduce. But if there's a rare event and we get a tetraploidization, so fourfold is easily divided by 2 when meiosis occurs. And so we can get gametes from those. However, they are diploid gametes rather than haploid gametes. And so those diploid gametes are unable to fertilize most of the individuals from the population, but they could self-fertilize in some plants, or they could fertilize another plant that was a rare tetraploid mutant. And so just like the pre-mating barrier we looked at in the snakes, this is by a biological species definition nearly instantaneous speciation, because you get that rare tetraploid. If it can reproduce and make a viable population, that population is nearly completely genetically isolated from its parental population. And so here we have example of 3 species that have arisen by this tetraploid speciation of Petiolaris and Anuus, or this, I guess they're both Petiolaris. So They've done it 3 different times and have made 3 different species in different areas. So that one is another kind of rare exception to a very rapid speciation event. And it turns out that when we look at the genomes of plants, this is actually a very common thing in plants. So polyploid speciation is responsible for much of the diversification and radiation of our flowering plants. It just happens very commonly. It's rare in other organisms because most organisms don't tolerate an extra copy of their genome. It's just genetically a really bad thing for them. But for some reason, plants have less of a genetic impact when they duplicate their genome. And so it's a really, really critical thing in the history of plant species. Okay, our last barrier that we're going to talk about is a post-zygotic barrier. So post-zygotic barrier is when mating can occur, when fertilization occurs and a zygote forms, but then something is going on with that zygote. The classic example of this is hybrid sterility. And you may already know about mules. A mule is the offspring of a horse and a donkey. So mating can occur, a zygote forms, it goes through development, is born looking healthy, but it's completely sterile. Right? So between those 2 species, there's not a pre-mating barrier, there's not a pre-zygotic barrier, but there is a post-mating barrier. And in other organisms, it's similar, but just not quite as severe, maybe not complete sterility. So for instance, tigers and lions can mate and have offspring ligers. Have you guys watched the Lion King? It's crazy. Anyway, there are ligers on that. And a liger will often have genetic abnormalities, meaning it doesn't survive well, or it's sterile, or just will not mate with other individuals. And so this epistatic conflict is a conflict between alleles. So alleles in the tiger community have been under selection within that community to make sure they're all kind of semi-compatible, with some rare exceptions, and in the lion community also. But when tiger alleles get exposed to lion alleles, there are often these genetic interactions that are just bad, and the organisms have defects, or they're sterile or semi-sterile. So an epistatic conflict is a great example of a post-zygotic barrier. And so there's actually a rule about this. Haldane's rule talks about how there's sterility between individuals when you mate. And you can look up Haldane's rule. I don't really care that you memorize it. It's a little bit more advanced in genetics class. You may have gone over it. Okay, so that's it. Remember those 3. We have pre-mating barriers, pre-zygotic barriers, and then the post-zygotic barriers. And so some species have only very recently diverged, lions and tigers. Zebras and horses actually can make a sterile or semi-sterile hybrid, also called a Z-donk. So there are many, many examples of where limited mating and zygotes can form, but there's still some fairly significant species barrier because the offspring just don't do well, are sterile or semi-sterile.